Hey there, I'm Emily Mong Douglas, and I'm with Louisiana Sea Grant, and I am really excited to have you all join us today for our webinar on HABs. That's part two of our two-part series on harmful algal blooms on the national stage and in Louisiana. We've got an awesome lineup of guest speakers for you today, and thanks so much for joining us. If you did attend part one or you really enjoyed part two and there are presentations in the mix that you really would like to see certain parts of again, please check out our YouTube page. You can check out Louisiana Sea Grant's YouTube at that link, that URL that's on the screen, youtube.com/Louisiana Sea Grant. The recordings have not been posted yet. However, they will be in the upcoming weeks, just as soon as we get all the captions edited and perfected to share with you all. So with that, on to the main show. We're going to kick it off with a little bit of information on the Atchafalaya Vermilion Bay System. We're going to hear from Dr. Beth Stouffer from University of Louisiana in Lafayette. We're also going to be hearing from Dr. Sibel Bargu-Ates and Dr. John White from Louisiana State University, and they're going to be sharing about these dynamic changes in deltaic Louisiana estuaries. And then we're going to finally hear from Dr. Matt Hyatt, Nicole Hammond, and Caitlin Turner, also from Louisiana State University, talking about Lake Pontchartrain. And then we're going to wrap it all up at the very end with a Q&A session with all of our guest speakers. So please feel free to share your questions in the chat throughout the webinar, and we will get to those questions at the very end with a Q&A panel style discussion. All right, so with that, I'm going to ask our very first guest speaker, Beth Stouffer, to start her presentation.
All right, you can see my screen? Oh, hang on. Nope. It's doing that thing again. Sorry, it reset. Give me 1 second. All right, hopefully y'all can see my presentation now, yes?
Yeah, looks great.
All right. Thank you so much. And thanks so much for the invitation to speak as part of this webinar. I really enjoyed having kind of a HAB focus in Louisiana as part of it. So I'll be presenting today on some of the work my lab's been doing in Atchafalaya Vermilion Bay System, kind of in the central western part of our state. And I just want to give a shout out to two PhD students, Andrea Jaggi, who recently defended, yay Andrea, and Julia Sweet, who's about this close to becoming a PhD candidate, who have done a lot of the work that I'm presenting today. I also want to just thank the rest of the lab here at UL, as well as some of our collaborators at LSU Ag Center, and then of course the funders for the work that I'll be presenting, some of it funded by Louisiana Sea Grant, but also Louisiana Water Resources Research Institute, or LWRRI, and National Academies of Sciences Gulf Research Program, Early Career Program Fellowship. So I loved getting the invitation to speak as part of this webinar because, and I think the idea of HABs in Louisiana is one that is a good kind of starting point. When you look at some of the maps, for example, of HAB distributions, the distribution of harmful algal species and kind of the toxins or the syndromes that they produce on the national stage, it's really interesting to always see kind of this little kind of like gap, this somewhat empty box where we don't see some of maybe the toxin producing diatoms or dinoflagellates or other groups that we see in kind of marine and coastal regions throughout the rest of the country. And actually, when I was just starting out here at UL, I submitted my very first Louisiana Sea Grant Omnibus proposal, 2015, tells you how long I've been here. And actually some of the reviews I got back were like, yeah, HABs are actually not an issue in Louisiana, and certainly not in some of the regions where I was saying I wanted to do some of this work. And I think that for a very long time, there has been this expectation or this thought that HABs aren't a huge issue here. But of course, over the last, several decades and years, we know that that may not always be the case. So whether it's work from Quaidorch in the 1990s, looking at toxigenic species of diatoms from kind of the estuaries out to the northern Gulf of Mexico, whether it's some of the work that Sabel and Matt and others are doing on cyanobacterial HABs in Lake Pontchartrain, which I think you guys will be hearing a lot more about today, or even, a recent kind of unknown mortality event in oysters that, we don't really have a good handle on what caused that. We know that, HABs are actually in Louisiana, coastal waters, if not estuaries. And we actually can see kind of a pretty big diversity of those bloom forming or toxic species. Some recent work out of my lab and with some great collaborators looked at community composition across 4 Louisiana estuaries, ranging from Breton Sound to Vermilion Bay. And when we look at just all of the genera that were observed across those estuaries, so nothing about, dominance or when and where, but we see that there are a number of those, what we consider more kind of marine HAB species, ranging from Pseudonychia, some of our toxic or bloom forming dinoflagellates, and even this little Rhapidophyte heterosigma, which has been in the news out in San Francisco Bay a fair bit recently. One of the other things that you can see in this data set is that we also have a lot of these freshwater, what we would consider freshwater cyanobacteria showing up in our estuarine systems, which makes a lot of sense for, I think, a lot of the reasons that we'll be discussing today. And so we know that these otherwise kind of freshwater cyanobacteria are problems because they can produce a number of different toxins. So different species produce neurotoxins, including anatoxins and saxitoxins, as well as hepatotoxins, so those affecting liver function, like the microcystins. And even this BMAA compound that's not necessarily a toxin has been linked to some pretty significant health impacts in humans. And so when we start to think about kind of cyanoHABs around the US, right, they're on these maps typically shown as occurring in freshwater, kind of inland bodies of water and the Great Lakes, and maybe a little bit in some of these estuaries. We know from some of the excellent work that's been done over the past couple of decades that cyanobacterial HABs, or cyanoHABs, as I'll kind of refer to them throughout the rest of the talk, are becoming more of a problem in estuaries because of some of the climate change impacts that we're seeing in those systems. So increasing temperature, increasing rainfall, or in our location, kind of at the mouth of the Mississippi River, right, increased floodwater, freshwater delivery, and then certainly high nutrient conditions all support dominance of cyanoHABs. And then, of course, we know in the Louisiana estuaries, some of the engineering, hydrological engineering and restoration that's being done also has the effect of, creating conditions that can even further support HABs. And I know the speakers after me will spend a lot more time on this aspect. So kind of jumping straight into the part of Louisiana's coast that we've been mostly focused on in my lab, let me introduce you briefly to the Atchafalaya Vermilion Bay system. I've kind of highlighted it here on the map. It ranges from kind of the Atchafalaya and Wax Lake outlets to Vermilion Bay towards the west. It's really 4 bays that are interconnected, and the system receives a lot of water, fresh water from the Atchafalaya River, which carries about 30% of the Mississippi River flow. So we have low, mean, and max salinities. This is what Vermilion Bay can look like on a summer day as the thunderstorms roll in. But if you get a salinity reading over about 5 in the system, it's notable. It's also a pretty shallow system, so 5 meters or less throughout most of it. And we know from some work that Nan Walker has done that winter storms, mixing events can both have pretty significant impacts on the biogeochemistry and some of the sediment dynamics in this system. So our most basic question that we've been trying to answer over the last few years in the lab are, are cyanotoxins, the toxins produced by these cyanoHABs of concern in this system? In order to address this, we focused in on microcystins. This is a hepatotoxin, again, produced by several genera of cyanobacteria. It affects liver function, but also has some other kind of health effects on primarily mammals. And for this toxin, there are both kind of ingestion or drinking water guidelines as well as recreational guidelines set by the EPA and World Health Organization. Since we're working in these estuaries, we're not so concerned with, ingestion as drinking water, but we are concerned with exposure of humans and animals, but also kind of the ecosystem level effects. And so to try and answer this question of, are cyanotoxins a concern in this system, we collected samples from 2016 to 2018 throughout the system, so these triangles on the map. And then in 2020, with support from the LWRI, we honed in on 3 nearshore sites, the circles on this map, to look at particulate, dissolved, and time integrative sampling approaches to these toxins. So why kind of break things apart in these different fractions? Well, we know that particulate microcystin, or the intracellular, so toxins within the cell, are kind of, I thought of as the more ecologically relevant exposure pathways for other animals, for food webs. So when a consumer consumes a cell that has toxins in it, right, those toxins can be bioaccumulated and magnified. We know that conditions like high temperature, or in the case of these freshwater cyanobacteria, increased salinities, in addition to physical disturbance like wind and wave activity, kind of bust those cells apart, make them maybe leaky around the cell membranes. And then that toxin becomes more abundant in the dissolved or kind of extracellular fraction. So Andrea Jaggi in my lab has done a lot of this work, taking samples, concentrating those particulate, and then sampling the filtrate for the dissolved fractions, going through the kind of standard, busting cells apart, extraction processes, and then running those on an immuno-based ELISA plate. These plates are tailored, so they are tailored using antibodies towards this ADA complex of the microcystin molecule. It's important to note that microcystin can take many different forms, that this ADA kind of moiety on the molecule is well correlated with some of the more toxic congeners of that toxin. We also deployed what are known as SPAT bags, which stands for solid phase adsorption toxin tracking devices. And in our lab, we make these out of just some embroidery hoops with Nitex mesh and a resin inside. And what these do is they kind of, they simulate what shellfish do, or what animals in the environment do, where the toxins absorb onto that resin, and they should be kind of held there until we go and retrieve the devices, extract them, and run them using the ELISA. We aimed for about 2-week deployments. Again, this was just in 2020, but of course, doing this work in South Louisiana during hurricane season means we had to be a little bit flexible. So some of the results that we're finding, one of the main takeaways is that, yes, we are seeing microcystins in this system, but they're very low levels. So the particulate, less than 0.1 micrograms per liter, which if you're familiar with the ELISA kits, that's what we're showing here. Sometimes is some of our data are actually below the detection limit, but with the particulate, since you're concentrating the particles and the toxin, you can kind of measure that. And in terms of the dissolved fraction, we see about an order of magnitude higher on average toxin. So yes, we're seeing very low concentrations of microcystins. Most of it in the system are in the dissolved fraction. If we focus in on the particulate side for a minute, we see that we had more positive tests. So when we were able to detect the toxin at these 3 nearshore sites. So, and this was across kind of all of the years. So Intracoastal City, this site 2, which is very close to Sycamore Point, and then Burns Point. And we know that these systems, especially over here, well, we think that over here in the western side, where you have Marsh Island kind of serving a little bit as a barrier to the Gulf of Mexico, we expect those areas to have kind of higher residence times, longer residence times. So supporting kind of, maybe these more stable conditions for supporting the kind of within the cell toxins. But we try to understand factors that contribute to toxin detection, so whether or not it was there. two variables kind of popped out. So this was using a logistic regression, and we see that there are higher odds of microcystin detection with increased silica, but with also decreased nitrate. And so this is a little bit maybe, we'll come back to this in a second, but the silica part especially clued us into, well, maybe it's not just the biogeochemistry, but also relationships and interactions within the phytoplankton communities in these systems that are, representing times when the cyanobacteria producing these toxins are outcompeting others. Interestingly, we never saw in this study the particulate microcystin at site 5, which was also the highest mean salinity, suggesting there might be kind of an upper limit for when we get kind of the cells with these toxins in them persisting. And finally, on the particulate side, the particulate microcystin did not really correlate with any of the biogeochemical or even physical factors we considered, but did have kind of a weak correlation with overall kind of chlorophyll and especially larger size fractions of chlorophyll biomass. Again, pointing to maybe there are some within the community interactions at play here. If we switch briefly to looking at the dissolved size, the dissolved fraction of microcystins, we never detected that at this BP site, which is closest to those freshwater inputs from the Atchafalaya and Wax Lake outlet. Although, again, this was a pretty limited study over just 2020. What's interesting is that the dissolved and the particulate toxins were pretty strongly decoupled, where we had a lot of, we had very few samples where we saw both. We probably need to look at this with more of a kind of time lag, which we do have the ability to do more of in the 2020 data set. But, we're seeing either particulate or dissolved and not often both fractions of toxin. And then finally, thinking back to some of those mechanisms we know to maybe move toxins from within the cells to outside of them, the dissolved microcystin concentrations were really only correlated with wind speed and silica. So again, we have that silica piece, which we'll come back to in a second, but the wind speed result was actually somewhat the opposite of what we expected, where we would expect winds in the day or two prior to, maybe enhance the release of microcystin from the cells. What we actually see is the opposite effect. And we think that that might be related to where we saw most of those dissolved toxins on the kind of westernmost part of the bay. So lower wind speeds kind of further increasing residence times, reducing mixing, and maybe supporting those cells that then get lysed or disrupted from other factors. If we stick for a quick second, though, on the silica piece. So many of you are probably familiar that silica is delivered from river sources into our estuaries, but really it's diatoms that are the main group within the phytoplankton who take that up and use it to build these lovely silica-based cell walls. And so this relationship between kind of silica and toxins indicating kind of cyanobacteria presence and toxin production really suggests that there's kind of these trade-offs or these shifts between diatoms and cyanobacteria in the system. And if we kind of take a really quick tangent into some work we did in 2019 following the massive Mississippi River flood that year, we see that shift from these black bars, so representing diatoms across multiple genera and kind of morphologies, to almost entire dominance by cyanobacteria in these gray bars. And these occurred really at the end of kind of following that flood period when there had been a lot of silica that was being delivered, but getting used by those diatoms. And again, we see this kind of relationship between increased cyanobacteria, but also that silica that's now available because it's not being used in the water column. Interestingly, we also see similar shifts in 2017 and 2016 at the same site, but much lower magnitude, just kind of small increases in cyanobacteria during this time. So I just want to wrap up some of the cyanotoxin work with the part about time integrative sampling. So those spat bags that were deployed at these 3 locations became really important for moving beyond those discrete grab samples, right, which represent a time, a place, and kind of that's it. And so, for example, at Burns Point, closest to, again, those large river inputs, we never detected dissolved microcystin from those grab samples. But you can see from this graph of the spat results, and I just want to note that these axes are comparable. There's a difference in unit, but we see that we actually did get detectable levels of primarily dissolved microcystin using these spat devices. So we really need to probably engage more with kind of looking over time at how these toxins are in the systems, especially for better understanding exposures and effects on animals and kind of entire ecosystems. And so to try and kind of answer the question of are cyanotoxins of concern in the ABBS, I think generally we can say maybe they're there. So microcystins at least are present at low concentrations and pretty widespread. At those low concentrations, they're probably unlikely to pose really acute health concerns. But again, this idea of kind of time integrative exposures, especially for kind of sessile organisms that can't move, makes it a question that we need to kind of follow up on a bit more. I think I have a few more minutes because I believe we started a little bit later. So I'm just going to share a few slides on some of the work that we're doing to try and link the microcystin or the causative organisms in these estuaries to one of those important sessile consumers that estuaries also support in Louisiana. And so I've talked a fair bit about microcystin. Microcystis aeruginosa is one of the species that can produce that toxin. It's a somewhat smaller cell, about five microns in size. We think of it as largely freshwater, but we know from all of the work that it can tolerate salinities up to about 15 or 20. 10 seems to be kind of more common. What's also of note is that it's generally considered a poor food item due to ratios of kind of carbon to nitrogen to phosphorus, as well as the lack of fatty acids. And then, of course, it can produce those toxins. Also in those estuaries, we have our lovely eastern oyster, Chrysostrea virginica, which we know can feed on particles that microcystis kind of fits within that size range. Similar salinity optima, and of course, I don't have to probably tell this crowd that this is an economically and ecologically important species. And so we know from a lot of freshwater research that some shellfish can selectively reject or choose not to eat microcystis, especially the toxic strains. So zebra mussels and golden mussels have given evidence for this. Yet at the same time, we see evidence for microcystins in Louisiana consumers, such as blue crabs and oysters. Some recent work looking at specifically kind of oyster feeding on toxic microcystis suggested that there were really no changes in oyster feeding when that toxic culture was fed alone or mixed with a more nutritious option. But I think notably, these experiments were conducted at pretty high salinities, around 15, which is pretty high, especially for Louisiana estuaries, who were conducted at really high kind of bloom level concentrations. And, clearance rate kind of tells you what's being removed from the water column, but there wasn't much done to look at where that went if the oyster was ingesting it. So some work that Julia Sweet in my lab has been doing with our collaborators at LSU Ag Center has taken on doing some more of these kind of oyster and microcystis feeding experiments. We've actually been focusing on a non-toxic strain of microcystis to really kind of hone in on these food quality, nutritional quality questions. This is work that Louisiana Sea Grant supported, where we've done these bialgal feeding experiments with non-toxic microcystis and a more nutritious but similarly sized diatom. Oysters were fed 3 diets, so all microcystis, all Thalassiosaria, or a mix. And we tried to better control kind of the concentrations as well as account for differences in cell biovolume. And then finally, from these experiments, Julia's been collecting data for clearance rate and also pseudofeces production. And I'm just going to wrap up with kind of these last couple points about, if you don't know much about oysters, they're kind of amazing, where they can ingest things, use them, digest them, and then put out a feces waste product, as you kind of expect. But they can also maybe encounter a particle that they don't want to eat kind of trap it and remove it from their gills and then put that out as pseudofeces. And very handily, they produce them on different sides of the animal. So it makes it really good for collection. And so what Julia has been doing in these experiments is documenting clearance rate. So these are the the 3 diets, 100% Microcystis, 100% Thalassiocyra, and then the mix. And we can calculate clearance rates on each species. And we're still working at this data, but it seems like there might be at least some reduction in clearance rate when Microcystis is offered in combination with something else, suggesting oysters may be selectively choosing the other species. But interestingly, we're not seeing a huge amount of difference in the amount of pseudofeces produced. So we would expect if they're selectively rejecting Microcystis that more pseudofeces would be produced. We do see a little bit of that in the 100% Microcystis diet, but again, doing some additional data analysis on this. And we want to go in and look at this 50-50 mix to see, using molecular approaches, if there's more Microcystis in that than we would expect. I think I've gone a little over time. Sorry, there's just so much exciting stuff to share. And so I'll just wrap up with, going back to some of the initial questions. Like, yes, we know that there are HABs in Louisiana coastal waters. There are many of those marine players that we often, think maybe can't make it in our estuaries. And I think, we do need to be doing a better job looking at those species, especially as our estuaries change. But we also know that these freshwater cyanoHABs are pretty widespread. So they're producing toxins in our low salinity estuaries, including the AVBS. Both abiotic and biotic factors seem to be controlling whether or not toxin is there and how much. And we think that, these cells, at least, plus probably their toxins, and there's some evidence for that, are making it into the estuarine food webs. What that means for, human health risk is one thing. What that also means for the health and ability for these important commercial and ecological species like the eastern oyster to survive and grow and thrive is another one that I think we need to be addressing. So I'll stop there, and I think we'll do questions at the end.
Great. Thank you so much, Beth. Next up, we're going to have Sibel start sharing her screen. We're going to have Sibel Bargu-Ates and John White from Louisiana State University sharing. While screen share gets up and running, I just want to give special thanks to the Sea Grant Network's National HABs liaison, Betty Stogler. She's located at Florida Sea Grant, and as well as Monica Wilson, also at Florida Sea Grant. These two colleagues of mine helped me conceptualize some of the things that I wanted to share in this HABs webinar series, and I just want to give special thanks to them. And especially Monica right now is on the line providing technical support for this webinar. So I just want to be sure to give thanks. And with that, I will get out of the way and let my guest speakers do the talking.
All right. Thank you, Emily. Good afternoon, everyone. Just to warn, if you were in the National Harmful Algal Bloom meeting last week, this is a very similar talk that I will be giving. And so just to warn, there's a couple of new additions, but it's pretty much the same talk. And since I will be talking, all the questions will go to John. So just to start with. All right, so we're going to be kind of looking for a larger perspective. Beth just gave a beautiful talk, giving into details of the toxicity of these cyanobacterial blooms. But John and I are working in the Louisiana estuarine system since 2008. And over the years, we have observed that changes in the climate and also all the implementation of coastal restoration activities are also influencing water flow and the nutrient dynamics. And the combination of these changes are impacting frequency and intensity and the spatial distribution of cyanobacteria blooms in these deltaic Louisiana estuaries. So we will be looking at a decadal perspective of these dynamics in multiple Louisiana estuaries for the rest of the talk. Coastal systems are exposed to a variety of impacts that originate from both natural and human-induced stressors. such as increase in external nitrogen, changes in hydrological cycles, and increase in sea level and water temperature, and the variability in wind strength. And all these stresses in many systems are yielding eutrophication of these systems that also results in increase in primary productivity and also changes in the physical environment. And they can all end up in resulting harmful algal blooms or hypoxia formations that we know have an environmental and public health risks. The overall productivity of Louisiana estuaries are physically and biologically impacted by nutrient-rich freshwater discharge from the Mississippi River. An increase in human population, so to be able to support the increase in human population in these land-water interfaces, people made a lot of alterations to this watershed. Levees is one example that was built by people to protect themselves from flooding. But while they were doing that, they were also altering the river flow and the reduction in nutrients and sediments to the wetlands, and ultimately causing wetland loss. And the accumulation and sedimentation of these elements back in the wetlands always is essential to regain these wetlands, and also offsets the natural subsidence. So in Louisiana, we have a lot of river diversions that reconnects the river with the marshes to be able to restore these lost coastal wetlands. These river diversions also have other ecosystem services, such as alleviating flooding, boost productivity, and reduce salinity. In some cases, this ecosystem service has a potential trade-off with the water quality of these systems. And we know that also the Gulf of Mexico waters are warming, and this warming also might influence the intensity and frequency of tropical storms. So, and because of both these climatic changes and anthropogenic activities, Louisiana estuaries are changing profoundly. And what happens is, with these changes, there is increase in the water temperature, there is an enrichment of nutrients and freshening of these systems, and the variability in nutrient dynamics. That kind of all yields to a somewhat optimum conditions for cyanobacteria blooms to occur in Louisiana estuaries. So cyanobacteria, and Beth mentioned it a little bit as well, is these are photosynthetic bacteria like land water. They does the photosynthesis like land plants do or algae in the water. And their bloom are becoming a global phenomenon, because especially in calm, nutrient-rich freshwaters and estuaries. And I assume in many systems like we are experiencing here, there are a lot of shifts in the dynamics that favors these cyanobacteria blooms. And one of the danger of these blooms, not only just the quality of the food themselves, but also they are capable of producing multiple toxins that can cause animal poisoning or human health problems. And in Louisiana, we see these cyanobacteria blooms in multiple estuaries. We have seen in Lake des Alamans, in Barataria Bay Estuary. We have seen in Lake Pontchartrain Estuary that has a lot of dynamics due to Bonnicaire spillway opening, as well as the Breton Sound Estuary, where Carnarvon freshwater diversion somewhat influenced the phytoplankton dynamics in the system. So the diversions are connected to these cyanobacteria blooms because they will bring nutrients that might potentially support their growth. But the link between the diversions and cyanobacteria bloom or their toxicity is not really as straightforward. So I'm just gonna probably take you a roller coaster for the rest of the talk while I give these variability that we have observed, especially in Lake Pontchartrain. So I'm gonna give you two estuarine examples that we have seen cyanobacteria blooms, but these estuaries are very different from each other in terms of their morphology, how they connect to the river, and the kind of how they show these cyanobacteria blooms throughout the time. Breton Sound estuary receives the river water from Carnarvon Diversion. And the reason why originally this diversion was built was, I'm sorry if there's a background noise, there's some kind of a party going on in the hallway. So the original purpose of the Carnarvon Diversion was to reduce the salinity to promote oyster production. This estuary is it's a salinity gradient. There's a freshwater upper estuary mixes well in the lower estuary with marine water. So there's a nice salinity gradient we see throughout the estuary. The receiving basin is vegetative wetland, and there's an annual entry of the river, which is a high flow compared to Lake Pontchartrain estuary that we see next. On the other hand, Lake Pontchartrain estuary is an open water system, and throughout the estuary, we see a low salinity. We don't see much of that salinity gradient the Breton Sound has. And the river input comes from the Bonacaire spillway diversion. And the purpose of that diversion was to prevent flooding of New Orleans when the Mississippi River water is very high. So we're going to start with the Breton Sound estuary. This estuary shows more kind of a seasonal distribution in terms of phytoplankton community composition. If you look at it in the springtime, we do see diatoms and chlorophytes, and that replaces with cyanobacteria in the summertime and also early fall with the increase in temperature and the lowering discharge conditions. When this shift happens, we see a corresponding increase in microcystin production in these summer and early months. But what was interesting when we did look at this system is presence of microcystin as well as the levels of them were higher when we were getting away from the Carnarvon Diversion. So what that tells us is during periodic high flows, potentially these cyanobacteria can be transported from upper estuary to lower estuary where the oysters are. And we know that cyanobacteria can adapt to low nutrient and moderate salinity conditions. So if that is the case in our system, they can sustain their biomass and create a potential risk for oyster contamination. This figure here is an unpublished work that my former graduate student, Emily Smith, and I were just kind of exploring. We exposed oysters to Dolichospermum, which is formerly known Anabaena. And we have seen that oysters were, like Beth was suggesting, sometimes can reject toxic food. They were not doing that when they were feeding on Dolichospermum. So there was a high rate of removal when they were fed with Dolichospermum. So obviously this is something to further look in the near future. Lake Pontchartrain estuary, as I mentioned, influenced by Bonacaire spillway, but in the past, the opening of this river diversion was not very frequent. It was maybe every 4 to 5 years, depending on the flood risk. But recently, if we look at it, it's almost an annual operation. In some years, like 2019, we have seen twice opening in the same year. When the diversion is open, here as you can see, there is a very nutrient sediment-rich freshwater comes and creates this very distinctive plume that separates the rest of the estuarine water when it's operational. So this is a period that I just want to kind of review what happens when the river diversion is not open. So there's a continuous input depending on the climatic condition, obviously, is the discharge that comes from the tributaries in the northern part of the estuary. There's a discharge from Lake Marupas. There's the leakage that continuously comes into estuary from the Bonacaire diversion. And then there's some tidal influence that might bring some saline water back in the estuary. When there is these conditions that introduce the nutrients in the summertime when temperatures are high. The water residence time is higher when you compare to the diversion operational years. So this is more kind of a calmer condition. Temperature is high, there's some nutrients coming in. We see a peak in cyanobacteria abundance. However, when the river diversion is open, when it's needed, then story changed tremendously. And as you can see, we see cyanobacteria bloom diversity in toxicity, abundance, and spatial distribution, depending on the timing of the diversion opening, as well as the duration of the open. So that's the roller coaster part that I will be kind of starting. So most of the times, the Bonaire diversion spillway is open in the spring times. And I'm going to introduce two springtime openings that we have seen in the past with two different outcomes. One of them is the 2008 opening. In this opening, there was a very distinct plume that expressed the phytoplankton productivity when there was some productivity going on in the rest of the estuary. When the diversion was closed after a month later, this plume collapsed, and then we start seeing primary productivity throughout the estuary. But what happened is, when after the closure, the light availability increased, turbulence reduced, we start seeing diatoms and chlorophytes. That shifted to microcystis when the conditions become more phosphorus limited, but still nitrogen is available. Microcystis is not a nitrogen fixing cyanobacteria, so it still needs nitrogen, but it's capable of storing some phosphorus internally, and it does better in lower phosphorus conditions compared to diatoms and chlorophytes. So when there is some kind of a nutrient shift, we also see a shift in the phytoplankton. And then now you ended up being both phosphorus and nitrogen limited condition. So there's a lack for phytoplankton response again until later in the summertime. When all the cold fronts and hurricanes come in, there's a potential for sedimented phosphorus to get resuspended in the water column. And those are the time we start seeing nitrogen fixing cyanobacteria like Dolichospermum and Cylindrospermopsis in the late summer and fall period. So in this scenario, we did see cyanobacteria in the early to late summer with different species succession. And overall, there was a succession throughout the entire phytoplankton community. And this is a figure from Roy et al. 13, and shows that during the opening, both nitrogen and phosphorus were available. They were both taken up by biological activity, and some of the phosphorus got sedimented. And then later in the summer, there was a benthic flux of phosphorus that we think supported these nitrogen-fixing cyanobacteria in that time. Another spring, early summer opening happened in 2011. And there was very much a high concern about this particular opening because there was the largest nutrient influx of any of the previous openings. We were expecting a huge harmful algal blooms and impact on fisheries. This is a comparison of 2008 and 11. So it's about, similar time period. It was open about a month, or the 2011 was a little bit longer than 8. And there was a lot of more river water coming in 2011. And if you look at the nitrogen and phosphorus, there was a lot of more nitrogen and phosphorus coming in 2011 as well. But if you look at this area, you see the productivity was not very different from each other. This is just the same thing cut out. So we have seen similar maximum chlorophyll A concentration with two different phytoplankton community. 2008 that I just shared, there was a phytoplankton succession that yields cyanobacteria bloom in the summer. But in 2011, we didn't see cyanobacteria bloom at all, even in the summertime. So we were just kind of scratching our head and looking at different parts. And this is another figure, I believe, from the same paper. Look at the northern tributary discharge and the Bonaire spillway discharge. It was interesting to see that the 2011 compared to previous years, the northern tributary discharge was lower. So that brought us a thought that maybe northern tributaries are providing seed population of cyanobacteria into the estuary. And when that discharge is low, maybe cyanobacteria is not as abundant or present in the estuary compared to sometimes that had a higher discharge that brought that cyanobacteria community into estuary. So that's something also to look into for sure. Now I'm going to switch from springtime to the winter time, which was a unique opening when you compare to other openings. 2016, we did have a Bonaire spillway opening in January. But because of the low temperature and the low light availability, we generally suppressed phytoplankton productivity, and there was no cyanobacterial bloom that resulted from that opening. And what happened is all the nitrogen came into the system, eventually left the system and transported to coastal ocean. And John tells me, and maybe he can say more about this, but the reason why this transport was not faster than it happened is because the wind kind of trapped the water in the estuary. And so there was more sustaining that nitrogen pool in the estuary before slowly leaked into coastal system. The second scenario to 2016 is not something we necessarily seen and have a lot of data, but if these temperatures are warming up, especially in late fall and early winter times, there might be a risk that we might experience in the near future. We know that microcystis and dodecaspermum can form dormant cysts on sediment surface when temperatures are low and nutrients are limited. If the temperatures are getting warmer and we are introducing these nutrients early in the year, we might provide an opportunity for these cysts to regerminate back in the water column. I had an undergraduate researcher that did some study on that preliminary work, which we didn't publish. I apologize for this kind of a weird graph. I couldn't regraph it before the talk, but this is 2019, November and December, and this is 2020, February and March sediment samples. And she looked at the phycocyanin concentrations in the sediments, and we have seen that in the late fall and winter, as well as early spring, we did have quite a bit of phycocyanin in sediment. And from this March sample, she brought it back in the laboratory and put the sediments in a container with the estuarine water, the filtered estuarine water. The control has no additional nutrients, and the blue one is the nutrient-enriched treatment. When they were sitting in a water above them with nutrient-enriched conditions, we started seeing increasing of phycocyanin in the water column. So this is something that we really would like to follow up and understand the potential of these early year cyanobacteria blooms if the conditions are changing in the near future. The last example I want to show briefly is the 2019 sample, 2019 opening. This was the only opening that we experienced twice in the same year. There was a spring opening and then the summer opening. And during the opening times, the volume of the water was, I believe, 28 cubic kilometer. And the estuary itself can hold 6 cubic kilometer. So most of that water came from the river, left the estuary, and kind of landed in coastal waters. And we collaborated with Rick Stump from NOAA, and he has these beautiful satellite images showing cyanobacteria blooms intensifying in these coastal systems, Lake Borgne and Mississippi Sound, during the opening time. and not so much seen in Lake Pontchartrain estuary. And when the second opening closed, that the second opening was ended, we started seeing cyanobacteria bloom formation in the northern part. With the changes in the wind strength and the direction, we started seeing spatial expansion of that bloom for the rest of the estuary as well. But what was most interesting about this year is we did not see species succession. We have seen coexistence of Microcystus and Dolichospermum together. Remember, in 2008, we did see this succession based on the nutrient dynamics and the availability. But in 2019, the temperatures were warmer, the system was freshened, both nitrogen and phosphorus was available, especially after Hurricane Barry. We have seen these peak in phosphorus. So there was no limitation of both nitrogen and phosphorus, and the temperature was high. So we have seen both of these species coexisting together, which has concerns because each of these species can produce, Microcystin is the common one that they can produce, the Beth was talking about, but Dolichospermum, can also produce neurotoxins like soxytoxin and onotoxin. And the only toxin we measured in this year was Microcystin, and it reached up to 7 microgram per liter. So that's pretty high compared to what we see in other times in these estuaries. So in conclusion, the climate change and all these anthropogenic activities has a synergistic impact on changes, intensity, frequency, and toxicity of these cyanobacteria blooms. And over the years, we have seen several factors being very important, kind of playing what the end product is going to be in terms of the bloom formation. Those are water residence time, timing and the duration of the river input, plume hydrodynamics, nutrient availability, tributary discharge, and weather conditions. So when the conditions are right, we may start seeing these blooms from the very beginning of the year, extended throughout the end of the summer or early fall. And one recommendation that we have done at the HAB meeting as well is when we are modeling or trying to forecast these models, we cannot focus only one issue at a time. It's really the collection of all these parameters together comes together to give a shape of these cyanobacteria bloom presence. With that, these are my acknowledgement. There are so many people involved, collaborators, students, technicians, and we have received multiple fundings from different agencies. So we are very thankful that we finally trying to solve the puzzle about these systems for cyanobacteria blooms. With that, I think I will end my presentation.
Awesome. Thank you so much for that. All right, we're going to move on to our third and final presentation. While Matt pulls up the slides, I'm also going to give a quick thank you to Melissa Castleberry, who's with our Louisiana Sea Grant communications team. She's always gives amazing support for all of our webinars. So I just want to give a quick shout out of thanks to Melissa. All right, Matt.
Is the screen good?
Yep, looks good.
All right, thank you. It's great to go last because Sibel did a wonderful job of explaining some of the motivation behind the work that we're doing here. And this project is in collaboration with the previous talk. Dr. Sibel Bargu and John White have been heavily involved in this talk this work as well. And we're trying to understand some of these physical controls on the phytoplankton community adaptations in Lake Pontchartrain, specifically related to some of those factors that was mentioned in the last talk. The spillway operations, the dynamics of the tributaries, wind speed, things like that. We want to marry essentially these physical drivers with understanding how these algal bloom dynamics occur. So of course, we're focused in Lake Pontchartrain here. We have this, it's a beautiful image of a algal bloom here on the North Shore, where a lot of these cyanobacteria, harmful algal blooms, tend to occur. And this project is a combination of both monitoring, so continuing some of those intensive monitoring programs within Lake Pontchartrain for the water quality, the nutrients, as well as sampling for the cyanobacteria and phytoplankton as well, but also to monitor the physics, so monitor the water speed, water level, monitor temperature, these types of things to get information on this aspect of the environment. So with that, we have a good team working on it, of course, and the two most important people, in my opinion, here are Nicole Hammond, a master's student working with Sibel on understanding the biological aspects, and Caitlin Turner here working with me on understanding the hydrodynamics of the system. And they are going to be splitting this presentation with me, so we'll hear from these two experts here in a moment. One of the major considerations that was mentioned in the last talk is this concept of the water transport timescale, or the residence time. It's been well documented that the time it takes water to move through a coastal freshwater environment is pretty nicely correlated with the ability of the system to remove nutrients. So specifically in this case, remove nitrogen. And it's also been well documented that that is correlated strongly with algal blooms. So a great example of this is in Lake Erie. You can see here an example of a algal bloom dominating much of the western portion of Lake Erie. And there was quite an intensive modeling and monitoring effort associated with this particular algal bloom right there. And what you'll see here on the left is outputs from a hydrodynamic model. So these arrows represent the water velocity in Lake Erie, and the scale here is about 10 centimeters per second. And you'll see here that in this region where the algal bloom was the most intensive, that is the region in which we had the highest residence time. These are essentially a measure of residence time. So water is staying in this region a very long time. It's circulating and it's allowing algal blooms to occur. And so we can say, I think, with some confidence that travel time or residence time, however you want to phrase it, is strongly correlated with the presence of algal blooms. And that's something that we're attempting to do here in our project, to make that connection. So we're this aspect of monitoring the physical drivers, this phytoplankton and water quality sampling, and our hope is that we'll be able to resolve both the conditions that give rise to harmful algal blooms or cyanobacteria blooms, and to identify both the temporal and spatial characteristics of those blooms. I would say at the moment we are in this phase. We haven't quite hit the equal sign yet, but we're making some good progress. So this is just an overview of where we are in the project. We've been doing some water quality, nutrient, and biological sampling in 2021 and a little bit in 2020. As we know, there were lots of challenges with fieldwork in 2020 due to COVID-19. We also collected quite a number of physical measurements throughout the system, and Caitlin will talk about that. And we have a numerical model, so a hydrodynamic model that we're setting up in the modeling software called Delft3D. And we've been able to create that model and perform a calibration, and we have some initial results on the circulation. And at the moment, we are working on quantifying these water transport timescales and correlating that with the water quality, nutrient, and biological sampling that's been performed by Sibel and John White's teams. So with that, I'm going to hand the presentation over to Nicole Hammond, who's going to talk about her biological and water quality sampling.
Yes, can you all hear me? Okay. So as Matt mentioned, I'm going to talk to you a little bit about the monitoring results and specifically about the biological and environmental data that we collected throughout the year. So as previously mentioned in Sibel and John's presentation, we have a really strong understanding into how the operation of the spillway contributes to the formation of harmful cyanobacteria blooms. So we're really interested in untangling those other physical drivers that may result in bloom formation when the spillway doesn't dominate the system. So 2021 allowed us to do that, and we collected monthly water samples throughout the year, and we collected them all across the Lake Pontchartrain estuary. And throughout the rest of this presentation, stations are going to be classified based on their hydrologic influence. So our stations in yellow are our stations that are influenced by the spillway. Our stations in orange are our stations that are influenced by tides. And our stations in red are our stations that are influenced by tributary discharge. And within our stations influenced by tributary discharge, we have our N1 through N3 stations, which is influenced by discharge from the northern part of the estuary. And additionally, we have station W1, thank you for that, W1, which is influenced by discharge from Lake Maurepas. So at each of these stations and sampling periods, we collected field measurements such as Secchi dis-dep, temperature, salinity, dissolved oxygen, and pH. Additionally, we collect water samples and process them for nutrients, suspended solids, chlorophyll A, phycocyanin concentration, which is an indicator of cyanobacterial biomass. And additionally, we analyze them for the cyanobacterial toxin microcystin. Slide, please. Okay, so to set the stage with some of the hydrologic inputs into the estuary, overall precipitation and total discharge followed seasonal trends with higher rates in the spring months. We saw increases in discharge and precipitation later in the year when we had a major environmental disturbance such as a hurricane, and this year it was Hurricane Ida. Additionally, we saw higher discharge from Lake Maurepas in comparison to discharge from our tributaries in the northern part of the estuary. And additionally, there was actually leakage from the Bonnet-Carré spillway that introduced fresh water into the estuary from the months of March into early May. So transitioning to now look at the environmental conditions throughout the year. So our water temperature followed seasonal trends, with warmer temperatures in the summer months and our coolest temperatures in the winter months all across the whole estuary. We saw strong spatial variability, or strong spatial and temporal variability in salinity, and these were predominantly driven by our changes in precipitation and discharge throughout the year. Overall, pH and dissolved oxygen showed no significant trends. And our spillway stations had the most turbid conditions, while our tidal stations had the least turbid conditions. So transitioning to look into our nutrient dynamics, we saw similar trends across time within our hydrologic influence stations, our division of hydrologic influence stations. We saw increases in bioavailable phosphorus and nitrate, and these corresponded to times throughout the year when our freshwater inputs were high. Similarly, we saw increases in this bioavailable phosphorus and nitrate later in the year when we had sediment resuspension, and this was predominantly from Hurricane Ida. We saw decreases in our bioavailable phosphorus and nitrate, and this throughout the year, and these decreases were attributed to when biomass or cyanobacteria biomass was highest, and this is representative of biological uptake. Additionally, ammonium was highest when our cyanobacteria biomass was highest, and silica varied across the whole year. So transitioning, we're going to look at our distribution of cyanobacteria blooms throughout the year. So first, we're going to start off with our spillway influence stations. So our PC to chlorophyll ratio represents our biomass on the left-hand side. So we saw blooms throughout the whole estuary in those late summer into early fall months. And we saw blooms throughout the year except at our station T19, which is the farthest from the opening of the spillway. What's really interesting at our D2 station, which is our station closest to the opening of the spillway, we saw that first bloom in that later summer, early fall, but we also saw a second bloom later in the year in the months of November and December. So transitioning to look on the right-hand side of the slide, we have our toxin data for microcystin. So overall toxin levels were below the recommended EPA level of 8 micrograms per liter. But what's just really interesting, at that D2 station, we saw extremely high toxin levels with an average of 41 micrograms per liter during that second bloom later in the year. So transitioning to look at our tributary influence stations, our stations influenced by that northern tributary discharge, so our N1, N2, and N3 stations. We saw overall higher biomass when compared to our station influenced by discharge from Lake Maurepas. And similarly, we saw those blooms in those later summer into fall months. For our toxin data, overall levels were below that EPA threshold, and we saw a very similar trend to biomass, where overall our toxin levels were higher in our northern tributary stations when compared to our station influenced by Lake Maurepas. And finally, to end with our stations influenced by tides, this tells a really interesting story. So our stations that have a stronger influence by tides, which would be our E3 station, we saw an overall lower biomass, and we actually didn't document blooms throughout that year. While our stations that have less of an influence by tides, so our E1, E2 stations, we saw overall higher biomass throughout the year, and similarly saw those blooms in the late summer into early fall. And our microcystin levels tell a similar story with thresholds below that recommended level. And additionally, we saw overall lower toxicity at our station that had a stronger influence by tides. And this may, and we can conclude that tides may serve as a control force on biomass and toxicity. So it's been mentioned throughout a lot of these presentations today, the impact of wind. So throughout this year, we concluded that bloom formation is really driven by those localized freshwater inputs, whether that be discharge, tributary discharge. But wind plays a really important role on a large scale. So wind speed and direction has an influence on the distribution of cyanobacteria within the estuary and into the surrounding basin. And additionally, wind speeds have the possibility to contribute to resuspension and alter light availability, or may result in the physical transport of blooms across that marine or across that freshwater to marine interface. It also has a really important role in driving those changes in residence time as well.
So Nicole showed us that cyanohabs in Lake Pontchartrain varied by hydrologic influence and wind conditions. And then Dr. Barku and Dr. White showed us that they vary seasonally. Using this information, we want to investigate the role of hydrodynamics on the spatial and temporal distribution of cyanohabs. So in order to do this, we created a two-dimensional hydrodynamic model of Lake Pontchartrain and Lake Maurepas. So the first step in creating this model was actually to characterize physical conditions using a field campaign and publicly available data. So the moorings that we set out, they're denoted by green stars, and they were deployed for about a year. And they had sensors that measure water velocity in both magnitude and direction, as well as salinity, temperature, and water level. So then next, as Nicole mentioned, there are 4 major tributaries. So we denoted those with red circles, and those are also influencing physical dynamics. And then lastly, there's a NOAA gauge where meteorological conditions can be obtained. As we heard, wind plays a strong influence. So using these data, we began to characterize the physical drivers of circulation in Lake Pontchartrain. So the field campaign was broken into 2 parts. So the first mooring deployment was done in October 2021, and that lasted until about January 2022. This initial deployment was done to find a period of stable conditions on Lake Pontchartrain, meaning no major storms or cold fronts, so we could begin to quantify what drives circulation and water level when you have those average conditions. So on the right, we actually have a time series of water level, salinity, temperature, and water velocity magnitude for each of the stations over our sampling period. As you can see, there's a box with a dotted line. We had an event that dropped water level about a meter at all of our stations, and as well as temperature about 6 degrees Celsius at all stations. So using meteorological data, you can see, if you look at the picture on the bottom left, there was a massive cold front that went through. So we saw that there was a really strong influence on events like that during on water level and hydrodynamics in Lake Pontchartrain. So that made it much more important to make sure we found a time period that had much more stable conditions. So if you look at the time series, you can see that from November 8th to December 10th, so that big black box, it's pretty stable in terms of water level, where you can really see the spring neap cycle. You can also see a lot more consistency in terms of magnitude. So using these baseline conditions, we were ready to create a model using the field data. So in a model, we have what's called boundaries, and those are where we specify a specific input condition. So one example of these boundaries is at the top. If you look, it's our tributary discharge. So in those areas, we physically put in a daily mean discharge, and then that was used along with an amplification factor that accounted for the distance between the river gauge and the actual mouth of the river into Lake Pontchartrain, to then inform the freshwater input. And then these were also forced with a salinity of 0.2 to represent freshwater. The next boundary that we forced was at the Wriglets, so that's in green right there. And there is where we probably do the most forcing, and that's where we put actual time series of data from the field campaign. So we put in water level from that period, salinity, and temperature, and then those will then, that data gets input and forces throughout the entire model, as you guys will see in just a minute. So this model was run for 33 days, and at each of the mooring locations that were on the first slide, those green stars, we put an observation point in the model. And what that basically means is it's kind of putting a fake mooring there and then giving us those same data that we got in the field that give us measurements of salinity, temperature, water level, and velocity. And this gives us a good way to quantitatively compare our results and validate our models. So the first model run I'll show you is the baseline conditions in Lake Pontchartrain that was created using the data from the field campaign. So this model right here is showing you an hourly output of salinity. Salinity is a useful tracer for understanding flow dynamics, since each of our hydrologic inputs have a specific salinity, such as riverine being pretty much 0.2 to 0, and then the tidal actually having that range of salinity that comes in through the Gulf. So you can see that the tidal signal is felt throughout Lake P, and then there's some areas that are impacted a little bit more by tributary discharge, and give you those plumes. And so now that we've gathered these baseline conditions, we wanted to see how the spillway opening would impact those dynamics. So this is the Bonnie Carey Spillway, as Sibel talked about a little bit. We know that it's open to relieve flooding pressures for Mississippi coastal communities. So on our right, we have a time series of spillway openings ranging from 1945 to 2020. So those are the majority of the spillway openings. So you can see that they really range in terms of how long they were open, the discharge, and that time period. So we chose a specific spillway event for kind of 3 reasons. It was 23 days, which my model was running for 33 days, so it made it fit within the model, as well as its maximum discharge being about the median discharge between all the other spillway openings. And it was a pretty recent spillway, so we knew that spillway opening, so the data was pretty good. So that bottom figure with the discharge over the day of the year, that was then forced at the spillway boundary in the model. So then using this data, we created a second model to investigate how the spillway affects water on Lake Pontchartrain. So again, we use salinity as a tracer. And as you can see, once that spillway opens, you get this really large plume that starts to take over, and it really dampens the tidal signal even. Sometimes when you get those stronger tides, you can still see the momentum, but the magnitude of water that's coming out is really driving dynamics in this area. And one of the things you can especially notice is these pockets that are just being trapped. So again, that goes with what Nicole's data was showing, is up at the North Shore, that salinity isn't really capturing right there. So there's a boundary that's just not getting impacted by the spillway, which suggests that there's an increase in the residence time there. So when looking at the spillway opening, like I said, you can see that there are pockets where exchange is being limited, such as the North Shore and actually a little area on that south eastern shore. And we compared this this model of the spillway opening with satellite data from Landsat 8 and their model of the spillway opening, and or their actual visual images of the spillway opening, my apologies. And you can see those areas, we are effectively capturing those regions that aren't affected as much by the spillway in the sense of the water getting there, but are affected more by being trapped by that sharp gradient. And so this is just a good qualitative check. It's promising because it shows that we're capturing major flow dynamics in zones that are impacted by the spillway opening. So we also wanted to review how our model is capturing hydrodynamics. So at the bottom, this is our baseline conditions. So similar to like an R-square value, we're looking for a skill score value. And it's similar to an R-square value in the sense that you want to be as close to 1 as possible, but greater than 0.85 is really good too. And so you can see that we're highly effective in capturing that water level with the riglets having a skill score of 0.96. And so our next step with this, now that we have our operational model, is to use a particle tracking algorithm to measure water transport timescales in our baseline model run, and then run models with varying hydrological inputs and wind conditions to determine how physical drivers impact water transport timescales and in turn the particulate matter such as cyanohabs.
All right, so thank you to both Nicole and Katie. Those were excellent summaries. I just want to close with this. We don't necessarily have any concrete conclusions. As you can see, much of this is very much hot off the press. But just as Katie mentioned, right, we're going to now move into quantifying these water transport timescales using this particle tracking algorithm. And we're going to see if we can make the connection, right, between these local changes in water transport time, where these regions where water is trapped or not trapped, and the prevalence of algal blooms as measured either by satellite imagery or by the monitoring that we've done in the field. So I'll leave it at that. Let everyone know this is, a work in progress, and we're happy to take any of your suggestions and questions. Thank you.
All right, big round of silent applause for all of our guest speakers. Okay, I'm going to now ask if you all can share any questions that you might have in the chat for our guest speakers. While you're formulating some thoughts, I do have a question for our guest speakers that came in during the registration period. We had the question of how are the troubles with HABs in Louisiana unique? In what topical areas would research be most useful to Louisiana?
I can start and maybe my colleagues can join me. Louisiana is unique in a sense that it receives, first of all, an input from such a large Mississippi River. And that not only influences estuaries, but also influences coastal waters. So we have this beautiful freshwater to marine continuum that phytoplankton can respond, different phytoplankton groups can respond to this same nutrient enrichment. So we might have these cyanobacteria in some location meeting with some of the harmful algal bloom species from marine systems. And when this organisms which is very adapted to these changing conditions can actually coexist at some kind of a threshold and might increase the contamination risk for organisms living in and also for human health. So in that sense, it's more complicated because of the diversion operations that we talked. It's more complicated because it's so vulnerable to climatic changes. And it's so unique in a sense that it receives tremendous amounts of pollutants and nutrients into these systems that these organisms have to respond somehow, adapt or no adapt, but coexist in some cases, and that can all relate to the health of the environment in general, as well as for us to be careful not to get those contaminations to our system. So I think it's an unbelievably interesting system to look at all these interactions together.
Just maybe follow up on what Sibel kind of ended with. So, well, two things. One, it's this amazing freshwater to marine gradient, which is going to be changing, right? I think that's the other thing. So I think in terms of questions that we need to be answering is, how does what we understand about the current situation change with new hydrologic modifications, climate change, sea level rise kind of affecting our estuaries. But I think the last point that Sibel made was really good as well. So obviously when we think about toxins transferring through, for example, shellfish to humans, like human health risk is a concern. But there's also, I think, a little bit of a void of, what are the effects on those animals too? And so I think we both presented some work with oysters. We know oysters are getting hammered from multiple directions in terms of whether it's salinity or, pathogens, in terms of understanding how the survival, the growth, the reproduction, kind of the stability of some of those ecologically and economically important species are impacted themselves, not just as a vector for human health, I think is still a pretty wide open area of research that we could address.
Great. On to the next question. Beth, how did you calculate a prime integration of toxins in the passive sampler?
Yeah, I think there were a couple of questions about the spat bags. I'm happy to kind of follow up with folks if there are specific questions. I will also happily offer my grad student, Andrea, who I think is on this to follow up as well, who really kind of got this method going. So the time integrated part is you put the bag out, you leave it, in our case, 6 to 15 days. You bring it back. There's some activation of the resin. There's some rinsing of the resin that happens. And then you kind of go through some extraction processes. At the end of the day, you take into account kind of the extraction volume, so the amount of resin you had in there, the extraction volume, as well as the amount of time that little embroidery hoop was out, and you take the concentration that you measure on the ELISA and basically correct for those other factors. If you remember that graph, you don't end up showing dots, right, or specific points in time and concentrations. And so the standard is to show kind of a bar, a line that represents what that spat bag saw over the course of that time period. So it's a little bit different in that, you can't say that, that toxin exposure was equally spaced out over the 15 days that was out. It might have been all at the beginning, it might have been all at the end. And so you really have to show that data differently. You have to take into account the amount of time that those bags were out. I will also just kind of make one point. Some of the work, a lot of this work has come out of estuarine HAB research in California. And we know that if these devices are out for long periods of time, you can't, so like a month or more there, that value may not represent just like loading and nothing being lost, and it might represent more of kind of an equilibrium concentration over that time period. I don't know that we've got a really perfect handle on that in our really kind of like high productivity, high everything else estuaries in Louisiana. But yeah, that is something to kind of keep in mind there.
Another question for you, Beth. What's the specificity of spat to bind toxins, and can spat be used to bind different types of toxins? And as a resin, as it is a resin, do chloride ions and background water influence it?
Excellent questions, I think from someone who kind of knows what they're talking about with some of this. And I see Sibel nodding too. So a lot of the work that has been done in the development of this spat approach has tried to quantify some of this. So HP20, the resin that we used, is one that's pretty broad in terms of there are multiple algal toxins that it will bind. Microcystin is one of them. Domoic acid, I was actually just triple checking which ones it binds. Microcystins, anatoxin, saxitoxin, domoic acid, and okadaic acid are all known to bind pretty well to the HP20. And there has been a fair bit for those toxins and this resin done in freshwater, brackish, and marine systems. So yeah, so we do have pretty good handle on that this resin is absorbing these toxins in these environments. I think there are some, I don't know, I've been hearing kind of rumors about some, other uses for the spat approach, whether it's, other dissolved compounds in water, like PFAS or other things, but, it is a resin that has specificity towards specific molecules. So Howard et al. 2014 is a really great kind of standard operating procedure. Nope, 2018, sorry, for these spat approaches. And I'm happy to follow up and probably put Andrea on following up with anybody who's interested in more.
Great, thank you. Thank you very much. A question that following up on a previous question that focused on what makes HABs in Louisiana sort of unique and special. I was wondering what lessons learned here in Louisiana do you feel could be applied in places outside of Louisiana? And this could apply to maybe specific findings or maybe techniques that you used or maybe even challenges that you overcame. This goes out to all my guest speakers. Or if you have specific challenges in your research, maybe you weren't anticipating that you'd like to share about, I think it would be helpful for our audience as well. I know there is a bit of mention about encountering hurricanes and just some of the climatic challenges that we deal with here in Louisiana. What other kinds of issues did you run into?
I can start a little bit. I think, most of the cases we do look at nutrient dynamics maybe more than the physical dynamics. And because the nutrients directly affects their growth and their toxin production per se, or the changes in the toxin type and so forth. So obviously it's super important. But I think our system shows how physical drivers can affect all of those things that nutrients are also affecting. So it's a good model, I think, when you look at different systems outside of Louisiana to maybe look deeper when you look at those synergistic interactions, when you look at the bloom dynamics. It's really not one way or one thing. It's multiple of those things. When I showed at the end a list of things that included biology, chemistry, and physics. And I think in that sense, our systems can be used in extreme kind of a nutrient enrichment, extreme of physical movements of hurricanes and so forth. All these things are maybe more enhanced compared to other systems, but it's a good model to show how those things interact with each other, which you can apply in different scales to any system outside of our system.
Sibel, I'll follow up on that. I think one of the challenges really comes, especially for Louisiana, when we do have hurricanes, when we do have river floods, there is substantial money available to monitor and go out and look at what's happening. Where we have a very difficult time over the last decade and a half finding money is in the years when that doesn't happen, when we have what's so-called the normal conditions. And then we've been really, really fortunate and grateful. Sea Grant has really stepped forward over the years and has provided us small pools of money that we're at least able to capture the data, so that the story we have now is really comprehensive because of we've been able to fill some of those holes. Otherwise, I think otherwise we would have a very incomplete story.
Yeah, I'll just echo the episodic, extreme, history breaking, event. I don't know, research opportunities that I think a lot of us take advantage of, and there are interesting questions there, but having good baseline pre-event, long-term post-event, in terms of some of the work that's coming out about accumulation of, for example, microcysteines and shellfish, is that, those don't just go away after a few days, right? That they can still be there for weeks. And so being able to, and who knows about longer, but being able to, so I think funding is one of the challenges, but you ask scientists that, and that's always an answer. But funding specifically for the base understanding, the fundamental understanding of how physical, biogeochemical, and then that biological within community, not to mention food web, effect or factors are leading to blooms, but also even like sub-bloom conditions where, some of these species don't have to be massively abundant to have toxins that we need to worry about. I think some of that is a real, remaining opportunity, if you will.
All right, well, excellent. Thank you so much for all of those perspectives and for providing your insights on all of these topics. And thank you for the shout out to Sea Grant as well. All right, so just a quick reminder to everyone, the recordings will be made available on Louisiana Sea Grant's YouTube page. In the upcoming weeks, you will get an email from me with that link, but in case you forget or you want to check out some of the other videos that we have available, there's a lot of different resources on the Louisiana Sea Grant webpage itself, but also on the YouTube page. So check that out. And if you'd like to provide some feedback, Melissa's going to be sharing a link for those of you who like to kick it old school and use a URL. For folks who like QR codes, you've got one in front of you. So please feel free to share some feedback about this webinar and tell us what you liked, what you didn't like, what we can do better, or if you have other topics that you'd like for us to cover in the future, even if it's not HABs related, if it's something else and it's coastal and it's sciencey. Yeah, we'd like to know. And okay, yep, that's my last slide. All right, so we're just going to leave that up. And thank you all so much for joining us today. And again, a big round of silent applause for our guest speakers.
Thank you, Sea Grant, for hosting this, and Emily, all of your hard work. It was really great to hear from everybody today.
Same. Thank you all for participating as well.